The first presentation is entitled Olive Leaves from Waste to Active in Pharmaceutical Micro and Nano-Formulations. That is presented by Professor Maria Camilla Bergonzi from the OLEAF4VALUE project. Camilla gained her PhD in Science and Technology of Medicinal Plants in 2002 and has a permanent position as Associate Professor at the School of Human Health Sciences of the University of Florence. She is the author of over 130 original papers in international peer-reviewed journals, has 1 patent, has written 3 book chapters, and is on the editorial boards of prestigious journals such as Pharmaceutics, Molecules, and Chem-Med-Chem. Camilla, please.
Thank you for the presentation. And I would like, first of all, thanks the organizing committee to allow me to present my research within the OLEAF4VALUE European project. OLEAF4VALUE means olive leaf multi-product cascade based biorefined. This is a 3 years project and will finish in June 2024. And the project is aimed at valorizing the waste of olive oil production to obtain products for the market application in the field of pharmaceutics, chemist, food, feed, cosmetic application. The coordinator of the project is NATAC. NATAC is a Spanish company, leader in the production, commercialization, research and development of bioactive compounds obtained by agriculture and also biomass. The partners of the project are 16 partners from 9 European countries. There are 3 universities, University of Florence with my group, University of Halle-Wittenberg and from Germany, and the Polytechnic Institute of Braganza from Portugal. Other partners are companies. OLEAF4VALUE covers the whole value chain. There are different work packages. The first work package is for the attention for the olive leaves, and then the valorization of the olive leaves, taking into account the possibility to obtain in the work package 2 the extract with high content of phytocomplex. The work package 2 is the biorefining and then the green extraction or extraction of purified compound or different extract with a high amount of constituents to produce matrices for the application in the work package 3. In the case of applications, there are many institute companies that improve enzymatic biotransformation of these compounds, or as my group that produce nano-formulations or microformulations for pharmaceutical application, food application, cosmetic application, and so on. The work package 4 is the package for the marketing application of these compounds. The UNIFI partner is a group leader in the research on natural compounds and extract from vegetable origins, and we apply a drug delivery system usually used for chemical compounds to natural constituents. We used, for example, microemulsions, nanoparticles, cyclodextrins, liposome, nanovesicles to improve some characteristic of vegetable compounds, because these compounds show a very high activity in vitro, but their application in clinical is reduced by some drawbacks of the compounds, in particular low solubility, low permeability, high metabolism. By application of these drug delivery systems to natural compounds, we can obtain and improve the solubility, permeability, stability. It's also possible for pharmaceutical point of view improve the targeting of action of these compounds and also reduce the dose and the side effects of these compounds. The role of UNIFI in the project, this is a pharmaceutical point of view, a little bit different from all talk of this congress, of this meeting, because I have pharmaceutical technologies, and then I have to prepare formulations to improve the quality of the compounds extracted by olive leaves. In this case, the role of UNIFI is in two work packages. Work package 3, in particular, I have to prepare nano-formulations to improve the technological aspect of the natural compounds obtained by olive leaves. And also, work package 4, I have to verify the influence of the formulations on some aspects, important aspects for human administration, solubility, permeability across the membrane, in particular in vitro intestinal membrane, and also on dissolution profiles. I will show you the results about the 4 formulations developed in my laboratory, in particular 2 nano-formulations, microemulsions and polymeric micelles, 2 micro-formulations, in particular solid dispersion and co-ground products. For this formulation, we performed in vitro release study, dissolution test to verify the influence of formulations, dissolution profiles of the constituents of the olive leaves, and also we tested the influence on permeability using in vitro tests, in particular PAMP-SA Caco2 cells. The matrices produced by NATAC, then by work package 2 of the project, are 3 matrices in particular. We obtain oleanoic acid at triterpenes, pentacyclic triterpenes, with high purity, about 97%. Also, we obtained an enriched extract of pentacyclic triterpenes. In particular, here the quantity, the amount, the percentage of triterpenes is about 70%. And also a polyphenol extract, an extract enriched in polyphenol contents, and in particular in oleuropein content. This is an extract with 40% with weight of oleuropein. The big problem for oleanoic acid of triterpenes is that these 2 matrices are completely insoluble in water. Polyphenol extract shows a good solubility in water, presents some problem about the stability, about the permeability. About the formulations, first step was developed microemulsions. Microemulsions is a system that contains water, surfactants, and oil. And in this case, we developed 6 formulations, 6 different microemulsions for oral applications, for oral intake. And with these formulations, we improved the solubility of oleanoic acid alone or triterpenes from 1 to between 1 or 3 milligram per milliliter, while the solubility of oleanoic acid alone is less than 1 microgram per milliliter. Then we obtain a very good improved solubility of these constituents. When we selected the constituents for pharmaceutical applications, for formulation, for human use, we selected the constituents between substances recognized as GRAS from FDA or compounds safe for human applications. In this case, we use different oil phases, capmul, nigella oil, isopropyl benzene, and also we used also compounds, in particular tenside, that improve the solubility, but also these compounds are penetration enhancer for improved permeability across intestinal membrane, and also PGP inhibitor. For example, TPGS is a derivative of vitamin E that is PGP inhibitors that can improve the permeation across the barrier. Macroemulsions, one of the aims of this project was to obtain a product for market applications, and microemulsions, taking into account these aims of the project, can be marketed or as solution, or liquid formulations, or without water, and then the oil and inside the formulations can be inserted in soft gelatin capsules, and then these are the 2 possible market applications of these formulations. The formulations has been characterized for stability, for physical chemical properties. Stability studies are related to storage, the stability study of the formulation, but also the stability of the formulations after intake in the body, and then the stability at gastric pH or intestinal pH and so on. We also verify by in vitro release study the release of a drug, in this case lanoic acid or the extract, from the formulations, because the release is important for absorption of the constituents. And we obtain different results. For example, in the case of lanoic acid, we have improved in vitro release, improved amount of lanoic acid released from the formulations. In the case of the extract, we obtain a prolonged release of extract from the formulations. We also verify the influence of the constituents of the formulations on the permeability of the lanoic acid, of triterpenes across intestinal barrier. We used PAMPA. PAMPA is a in vitro assay that is a system that contains a donor receptor compartment and an artificial membrane derivatized to mimic, for example, intestinal membrane in this case, but also skin, blood-brain barrier, and so on. In this case, we monitor also the influence of formulation of passive permeability, no active mechanisms are involved in this system. And for all formulation, we obtain a very high improved permeability with respect of lanoic acid or extract alone. There are two matrices impermeable across intestinal barrier. Other nano-delivery is polymeric micelles. This is a nano-formulations. To produce nanomicelles with polymeric micelles, we tested different amphiphilic copolymer. In particular, good results are being obtained with poloxamer or pluronics. These are tenside used for human applications. Are copolymer between ethylene oxide and propylene oxide. It is in the micelles we also insert as a polymer TPG, a derivative of TPGS, a derivative of vitamin E to improve antioxidant properties of the formulation, but also to improve the permeability across the intestinal barrier. We selected the formulation, the composition, considering the physical chemical characteristic of the micelles before but also after freeze drying process, because the freeze drying process is a method to improve the stability of the formulation, because we eliminate the water as eliminated, and then we have improve of stability during the storage of the formulations. We have good results with the pluronics L121, pluronic P180, and also TPGS. These polymeric micelles are able to load oleuropein in the extract of polyphenols produced by NATAC, about 50 milligram per milliliter. We obtain a good release, a prolonged release of oleuropein from the micelles, and also improve the permeability as demonstrated by PAMPA assay. We also, in this case, study the stability during the storage of the formulations with water and without water. Then we have two possible market applications. This aspect is very important, the storage stability for formulations. We obtained that freeze-dried product improve the stability of oleuropein. For this system, also, we study the influence of formulations on permeability by using a cell method, in vitro cell method, in particular Caco2 cells. This is a system similar to PAMPA, but on the membrane there are the cells. In particular, these Caco2 cells are colon carcinoma cells that differentiate in a monolayer that mimic the intestinal barrier, because the cells presents the brush border layer. Also, in this case, by analyzing the pH value and then the permeability coefficient of oleuropein without or in the presence of the micelle, we obtain a good improvement of permeability across the cells. In this case, we monitored not only passive mechanism, but also active mechanism involved in the formulation. About the solid formulations, we developed the solid dispersion and also co-ground product. We selected the formulation thinking about a possible market application, then a possible scale-up of the formulations. Solid dispersion is a dispersion of hydrophobic compounds, such as oleanoic acid or the triterpenes extract, and hydrophilic carrier. We tested a different carrier, we tested a different method for preparation for solid dispersion, and we obtained a very good results with two poloxamer, P407 and P188, and gamma cyclodestrins. During the preparation of solid dispersion, we obtain an amorphization of oleanoic acid, and this amorphization was proved by different technique. For example, X-ray is possible. For X-ray, it's possible to observe here the crystallogram of oleanoic acid inside a solid dispersion. These amorphous systems, and the solubility was increased until about 200 microgram per milliliter. Very good results and improved the solubility. Also, thinking about in the consideration the possibility to produce pharmaceutical formulations for market application, we prepare, in this case, hard gelatin capsule. Then we fill the hard gelatin capsule with solid dispersion, and we perform the dissolution study. And we can observe that with respect the dissolution profile in black, this line, without a formulation in non-solid dispersion, all solid dispersion improve the dissolution profile of the drug, of the molecules. This is the results for the extract of triterpenes, and we can observe that this is extract alone, a very low dissolution profile. This is an increased dissolution profile of extract due to the presence of solid dispersion, then the hydrophilic carrier. Also, pump assay demonstrated that we have improved permeability, passive permeability of both oleanoic acid of extract. The last one formulation is a co-ground product. The co-ground product was obtained by mechanical activation, produced in a ball mill. This is a mill for laboratory scale. And inside the mill, we insert oleanoic acid and a hydrophilic carrier. We tested different carrier with different condition of milling, and we have very good results as increased solubility using sodium cholate. In this case, we obtain a solubility up to 800 microgram per milliliter, with respect to 1 microgram per milliliter is very good results. Also, in this case, the energy applied by the mill produce amorphization of the crystals of oleanoic acid. It also improved the solubility by the van der Waals hydrogen bond interaction between carrier and drugs. In this case, we can observe that oleanoic acid alone has a needle-shaped crystal. Needle-shaped crystal also present in the physical mixture without energy, without mill. In the presence of co-ground products, we observe amorphization of the oleanoic acid. No needle crystal are present. Also, in this case, we prepare hard gelatin capsule, and we obtain the best results of all formulation developed until now, because we have a very high amount of oleanoic acid released from the formulations, from the co-ground product, with respect to oleanoic acid alone. Very good results for future next in vivo study. In conclusion, for pharmaceutical point of view, we have developed different micro and nano formulations, about 6, in particular 6 microemulsions, 6 solid dispersion, 2 co-ground products for triterpenes fraction, obtained from the olive leaves, 1 polymeric micelles obtained from for polyphenol extract delivery. We optimized the formulations, we improved also solubility, dissolution profile, and also in vitro permeability. And for lack of time, I didn't show the results now, but also the formulations can improve in vitro activity of the single compounds, in particular, for example, antioxidant activity of linoleic acid, or the inhibition of the extract on lipid accumulation inside the cancer cell, for example. Then we also observed an improved of biopharmaceutical pharmacological activity, in vitro activity, due to the presence of the formulations. I conclude and I thank you for your attention.
Okay, any questions on the floor?
Yeah, thank you for this interesting presentation. It's a bit different than what we're used to. So my question is a bit more close to what we do. The compounds that you extract from the olive oil leaves, are they typical for the leaves only, or are they also present in the olives?
It's typical of leaves, but also in oil fruit, and also in other part of the plant, yes. Not only leaves,
Okay.
also for fruit.
Okay. And
Other species of plant, not only olive.
no, but you extract it from the leaves, right?
Yes, Natac extracted these compounds from leaves,
And
but these compounds are also present in the fruit, for example.
yeah, okay. And how much do you extract from the leaves? Because I can imagine there's still a residual part of the leaf that you cannot use.
I don't know exactly
Okay.
percentage of from leaves to extract.
Yeah, okay, you really
The
focus
yield
only
of the
on
process, I don't know.
Yeah, okay.
Is Natac.
Okay, that will be in our interest, of course. Anyway, thank you.
Yes,
You're
and one of the following presentations will talk about actually the extraction of phenols from the pomace as well, right? So,
And they know the yield of the process. How many leaves to obtain. This is a problem for the project, the European project. Optimize the ratio, the extraction ratio.
And in terms of exploitation of the work you're doing, for exploitation by pharmaceutical companies and food industry, and what are they most interested in from you? What can be most
There
exploited?
are different problems. The cost, the scale up, and the stability of the formulations. I think that in particular Natac is interested in solid formulations. For example, solid dispersion or co-ground products, because it's more easy to obtain these products and also more stable on the market. For the market is the best the hard gelatin capsules, for example, not soft gelatin capsules because they're very expensive. Mm-hmm. Then better solid formulations. Liquid, the problem is the water, mm-hmm. For the stability of the preparation.
But is it more food supplementation? Are we talking here?
For food supplements, yes, because for drug there are many steps, about 10 years of the study, different in vitro, in vivo, clinical, and so on. Very expensive process. I think it's better for food supplements application.
Okay. Well, thank you.